Hello, and welcome to Field Lab Earth, the podcast that's all about past and present advances in the fields of agronomic, crop, soil, and environmental sciences. Today we'll be talking to Dr. Zoë Migicovsky and Dr. Carsten Pedersen about Genome-Wide Association studies in apples. What is a Genome-Wide Association study? How can it help apple breeders identify genes for desirable traits? And how does that knowledge get your favorite new varieties onto your shelf? Answers to all these and more coming right up. I'm your host, Abby Morrison. Let's talk about science. Hi, Zoë and Carsten. Welcome to the show. How are you guys doing today?
Great.
Yeah, I'm fine.
Awesome. So Zoë completed her undergraduate degree in biology in Nova Scotia at Acadia University, her master's in biology at the University of Lethbridge in Alberta, and her PhD in biology at Dalhousie University. She's currently a postdoctoral fellow at Dalhousie University in the Faculty of Agriculture, where she works on two projects, one on grapes and one on apples. Carsten works as an associate professor at the University of Copenhagen in Denmark, where he teaches bachelor and master students in genetics and genomics. It's there that he did his studies in agronomy in the 80s and finished his PhD in 1995. He's also worked abroad as a postdoc in Germany and Australia. His work is focused on interactions between plants and pathogens, but he's more recently moved into apple research. So, yeah, welcome to the show. So glad to have you. Right up front, most important question of the show as we are talking about apples today. What is your favorite kind of apple?
I have a couple favorite apples. I work on a pretty diverse collection of apples, so it gives me a chance to taste and try a lot of them. But definitely one of my favorite commercially available apples is Sweet Tango, and that's from the University of Minnesota breeding program. And it's actually offspring of Honeycrisp, which people are generally more familiar with, and it's got a really great flavor and texture.
Nice. Carsten, what about you?
Yeah, my favorite is, or one of the favorites is Elstar. Elstar is a Dutch cultivar, but it actually has a Danish ancestor. One of the parents is Ingrid Marie. It's a Danish grown cultivar. And Elstar is also, I think, has a good balance between sweetness and sourness and a great aroma. And it's also crunchy to eat.
Sounds wonderful. I've never had that one myself. I'm actually allergic to raw apples. I think it's like like a birch allergy or something that somehow got into apple. But I can eat them if I microwave them for like 30 seconds first. So I am partial to Kanzi apples. They hold their crispness after microwaving, which not all apples do. So I like that. They're nice and sweet. So we're obviously talking about apples today, but specifically kind of apple breeding. So let's just get some background information and terms identified so we can have a more efficient conversation moving forward. So let's just start with the basics of how are apples bred and reproduced and grown today. I mean, obviously, in America, we've got our Johnny Appleseed story of just chucking seeds everywhere until they pop up. But that's not really how it's managed today. Can you explain that process?
Yeah, I can start. I mean, apple breeding is relatively simple, I will say, because you can breed new apples just by crossing two different cultivars, and then you will probably have to select among many, many seeds and small seedlings you get up. And the problem here is, of course, that to look at the apples, you have to wait several years, like 5 years or more, before you have apples on a new tree. So it takes a lot of time. But in principle, it's quite simple. I mean, apples are self-incompatible, so they will not self themselves. When you get seeds from a tree, or you take the kernels out, then there are progenies from a cross, so the two different parents. Unlike many other plants, which you can self, so they are getting inbred lines or so. But then, of course, when you have a good apple, find a good seedling giving a new combination of the parents, then to keep that, you cannot self it and take the seeds because they're all different. So then you have to propagate it in another way, and that's where you do the grafting. So you take part of a branch and put on a rootstock, and you get many, many trees from the same original plant. if you need like get in a trait from the more wild type of apples, which are maybe not having so good apples, but they have some resistant traits or so, then you need to do back crossing. So you make your cross between your cultivar and then this more wild apple with good resistance against some pathogens. And then the first generation you have to cross again with your modern apple, and then you have to continue and to do this back crossing to the good cultivar for several generations. And then you can see each generation might be more than 5 years or so. So then it becomes really long to get in a good trait from a wild apple into the modern cultivars.
That's crazy. That's kind of like cheese or wine where you have to wait for it to age, and then it's like, only then will you know if your experiment worked from like 5 years ago.
Yeah, yes.
Talk about patience for your work. So one of the things we're going to be talking about today that aims to help with speeding this up a little bit is called a Genome-Wide Association study, and we're going to talk about that a little bit more in depth later, but can you kind of just give a brief overview of what that term means?
So to perform a Genome-Wide Association study, what we do is we collect genotype data from a whole bunch of diverse accessions. So in this case, they were all different apple accessions. And then once we have that genetic data, and we've also collected phenotype or trait data from those same accessions or same plants, then we can correlate the two with each other. And when we correlate them with each other, we perform genetic mapping. And the method that we're using, because we have a whole bunch of diverse accessions versus progeny from a single cross and a whole bunch of siblings, when we use diverse accessions instead, we're performing a genome-wide association study. And so it's a method of genetic mapping that we can use to correlate genetic data and trait data with each other in order to see which genetic markers are the most predictive of that trait.
Nice. And then one other quick term that might come up is just what a volatile is, or a volatile.
Yeah, volatiles are these compounds which are easily getting up in the air, so to say. So you can sense them, you can smell them, and in apples there's a lot of these fruity volatiles compounds like esters or alcohols, aldehydes, which are, so when you eat an apple, then you can actually smell it, and if you drink apple juice or so, and there are plenty of them and many different types. And that's what makes the aroma to the apple. So you can distinguish different cultivars just by the aroma. I mean, maybe not if you have to be trained to really say, okay, this is the cultivar, but you can say, okay, some have a better taste or aroma when you eat them than others. So that's one of the traits we were interested in to study further here.
So you were looking for kind of some specific traits to help find them across your Genome-Wide Association study. Can you tell me what exactly you were hoping to pinpoint in your research?
I was hoping to find something for the aroma, and otherwise we had a lot of data, and I was really not knowing what to expect to get out of it. So from this Pometum collection, we had recorded over the years many traits connected to the trees and the fruits, and one of these where we actually found something was this harvest date or earliness, how early the apple cultivars is, but there were many other traits where we didn't find anything actually. So of course we were hoping to find something connected to important traits, because that's where, if you are a breeder, you would like to have DNA markers which you can use for select for the most important traits. And among these is also disease resistance. And we had some measurements, we have some recording data for about how resistant the different cultivars is, but it's not so accurate. And some of these traits are also difficult to handle. There are many genes which are involved in the environment, and so on. So we didn't really find anything for that.
And you mentioned briefly the Pometum. And this is like one of my favorite parts of this research. So can you just tell us, what is the Pometum? What is it like to work there? What does it look like? Just tell me everything about it.
Okay, the Pometum is like an orchard, an apple and all other fruits as well. And there's just like in one section we have 2 apple trees of each of the cultivar standing there in long rows. And then you can walk and see on small signs what they are, and if you are there, a little earlier than now, but here in the autumn, in September or so, you can go and taste the different apples. It's actually quite fun. So it is a gene bank, so to say, where we keep the different cultivars. So if somebody are going to use them, they can come here, and they can also, private people can actually order branches for grafting. So if they want a certain old cultivar in their garden or so, they can order the branches and then make their own trees.
That is lovely. I mean, I already knew it was an orchard and I was excited about that, but I did not know that you could order branches. So that's really, really exciting too. So the Pometum is where you were collecting phenotypic data, which, as a reminder for listeners, phenotypic is more just what you can observe with your eyes or other senses, as opposed to the genomic data, which is more tied in with, the actual DNA. So you guys were collecting this data, you were using historical data, obviously had a wealth of information from the Pometum already. But then you also were pairing this with the genotypic data for this Genome-Wide Association study. Zoe, can you tell me more about how you accomplished that work and compared the two?
Yeah, so the type of genotype data that we used in this study is generated using a method called genotyping by sequencing, or GBS. And what that does is generate a whole bunch of short reads of DNA sequence. And so we take all of those short reads and we align them to the reference genome that we have for apple. And so after we've done that, there are regions of that read that we've aligned to the genome that match up, and there's some that don't match. And when we have a single base pair or nucleotide that don't have the same sequence as the reference genome, that's called a single nucleotide polymorphism, or SNP. And so those are the types of genetic markers that we used in the study, our SNP data that we generated using this method. And so we're only interested in regions of the genome that differ across the accessions that we've genotyped, because we're interested in correlating traits with genetic markers. And if all of the genetic markers have the same sequence as the reference genome, then there's no variation there to correlate. So we're reducing down our genotype data to only things that differ.
Okay. So it's kind of like if you have a bunch of apples that are all, for example, everything about them is completely the same except for like the color of the skin, then you would compare the pieces of these little snippets of DNA one by one until you find the DNA that's different between them, and then you'd be like, well, that must be what determines the skin, because that's the only thing that's different. Am I sort of in the ballpark here?
Yeah, so that's basically what we do, except that we have all of the genetic data and it's all being compared at once. And at the same time that we're comparing it, we're also correcting in our model for things like relatedness among the accessions that we have in the study as well. But yeah, we perform those correlations, and the genetic markers that are the most strongly correlated with the trait of interest are the most predictive of the trait that we're interested in, and the most likely to be either candidate genes or nearby to candidate genes.
Wow, that sounds really hard. And something that you guys mentioned in your worksheet is that it's also tricky because sometimes it's just like, oh, it's related to the weather or like some other environmental factor that may not have anything to do with the actual DNA or traits or, what have you at all. I mean, the trait itself, the way that it manifests. So can you talk about that a little bit too?
Yeah, it's correct that we have the environment which is influencing the trait or how it comes through. But the other thing is also that the trait itself can be made up of or influenced by many genes. So like the fruit color we talked about here, it's a relatively simple trait. So one gene has a really big impact on that one. But many other characters are influenced by many genes with just a small influence, and then it's really much more difficult to find the associations which are really coming well through in the analysis.
Hi everyone, I hope you're enjoying the show. Interested in learning more? Zoë and Carsten's article, Genome-Wide Association Studies in Apple Reveal Loci for Aroma Volatiles, Sugar Composition, and Harvest Date, published in the Plant Genome, is always freely available. You can find a link to it in our show notes. Let's get back to the show. So then, moving on to the results, what did you find?
Yeah, we find different things. So we just talked about the color, and that was like the very first thing to look at, because that was already done before by other people. So it's like a proof of concept that we could use our collection of trees to find the association for the color on the fruit. And so it was not a new result, but nice to see. And then among the other things, what we found was this harvest time point, and that was, I think, when we saw that first time, it was also new for me. But Zoë, you can maybe explain that you already have seen that in your group from the same result actually.
Yeah, so that's a result that we previously identified, but I think the work was ongoing at around the same time. And that was a result that we also identified using another germplasm collection similar to the Pometum, but located in the US at the United States Department of Agriculture. And subsequently, it's also been identified in a Canadian germplasm collection, and then also in a study that used germplasm collections from across Europe as well. So it's been validated a few times now in different collections that include all different kinds of apples. So it seems to be really strongly found across many different diverse collections of apples as a good candidate for ripening time or harvest date.
Anything else?
Yeah, so we can talk about the aroma compounds. That was where we had many expectations. And so the aroma was measured by a group here at Food Science in our university, where they have a good setup for measuring all these compounds by gas chromatography, mass spectrometry. And so they have a setup, so they measure the volatiles, which comes up if you blow air through this apple juice, and then it's collected from the air above the apple juice. And we measured in this study about 50 different compounds. But when we do the GWAS, there is really a lot of, you can say there's some noise, and there's not so many really nice peaks coming up showing that here you have a good association. Actually, we only find one which I find is really reliable. That's on a chromosome 2 of apple. And it's also something which other people have seen before, that there is something in this region on the top of chromosome 2 influence these, especially the acetate esters, which we see. So, well, it's challenging to find good associations for aroma, but we have found something.
Nice. And just for anyone unfamiliar, GWAS is the Genome-Wide Association study, just that little acronym. And then you were also looking at sugar types. Can you tell me, what does that do within an apple, and what did you find?
Yeah, so in apple we have actually mainly 3 types of sugars: sucrose and glucose and fructose. And the difference is that when you have sucrose, it can be split up into one molecule of glucose and one of fructose. And we find that there's, like, some cultivars have quite a lot of sucrose and not so much fructose and glucose. And then there are others which are really high in fructose and glucose, maybe not so much glucose, but much less sucrose. So we can see there's like differences. We can even talk about like 2 types, and then also some in between. And in our GWAS, we then find an association on chromosome 1 to this trait, to the balance between having high sucrose or having high fructose types. So that one aspect of this could be that if you want to ferment the apple juice to cider or apple wine or so, then there are differences in how fast the yeast is actually fermenting it and making the alcohol. The type of sugar has some influence on how fast the yeast is making alcohol and fermenting it. So, and actually, like slower fermentation may improve the aroma in the wine or the cider you make. So maybe it's a good idea to use some which are high in fructose for fermentation and have less sucrose and glucose.
Sure. You have to wait at least 5 years to find a new cultivar, and then you turn it into wine and have to wait even longer.
But about waiting, and that's where our data can be used in the future, because one application is to use to make DNA markers from these small DNA variation we find in the genome associated to the traits. And then you can use and select already when you have a very small seedling, and can, with one leaf, you can isolate DNA from half a leaf, and then you can screen and see what is the potential, what can we expect to get from this little apple tree in the future.
Sure, yeah. I mean, that takes your wait time down by, years. That's huge.
Yeah, yes, it does.
Yeah, one of the things that's cool also about the work that was done using the different kinds of sugars is that by making more accurate measurements that actually look at what different kinds of sugars there are, we're able to detect new associations that would have been missed if we just looked at overall sugar content. And so that's something that was really exciting in this study that we hadn't picked up on before in previous work.
Totally. Yeah. And that's kind of the advantage of pairing phenotypic with genotypic data is, you can taste an apple and say this is sweet, but unless you're very, very, very good, I'm imagining you probably can't really tell the difference between fructose and sucrose and glucose, just by eating it. But then you get that advantage of the genotypic data. So, yeah, we've kind of touched on some of this then already, but why? What are some of the very practical implications of this work, or why is this so important for breeders, people who are just growing apples, the general public looking to just purchase apples commercially? How does this all affect them?
I think ultimately this kind of work is useful for helping apple breeders accelerate and more efficiently select for the traits that they're interested in across the apples that they're breeding. And so the ramifications of that for farmers or for the public is that if we can accelerate the breeding of new apples, and if we can develop apples with new tastes and flavors and desirable quality attributes, then that's going to have a positive impact on people who are able to then purchase new kinds of delicious apples at the grocery store as well. And so making that process more efficient and allowing the breeder to more accurately select for what they're interested in is going to be able to benefit the apple breeding more generally, and then the release of new apple cultivars. And a lot of the traits that were measured in this study are very difficult to assess. Like you said, you can't just take a bite of the apple and necessarily know which aroma compounds are included in it. And so if we can allow breeders to select for those traits that are going to overall contribute to a desirable apple through genetic markers, then that's a lot more cost effective and efficient than having to grow up the trees and run all the apples through these kinds of analyses one apple at a time.
Totally. Absolutely. Another thing that we've briefly touched on a little bit is future research. So what are the next steps? Where are you guys going with this research, or just this field of research in general? What are the next steps to be done?
Well, I will try to get funds for continuing working on the aroma side. So I have recently applied for a new project on going more into detail with aroma, where we will try to do it better than this way, having more cultivars to look at and better measuring of the aroma content. And we would also like to actually add on top of that the sensory studies, where we not only measure the different volatiles, but we also look at what is actually their influence on the aroma as we perceive it when we drink apple juice or taste an apple or so. And it's very complicated because we need, trained panels of people which will then taste and describe, and you have to relate that to the different compounds because it's so complex when you take them all at a time. So it's very complicated, but I hope that we can find more associations to these traits to better describe them.
Yeah, like Carsten, I'm definitely interested in continuing to both genotype and phenotype diverse apple collections so that we can improve our accuracy of genetic mapping using genome-wide association studies and help develop genetic markers for apple breeders using more diverse traits as well. Of course, the apple breeder is always going to have an important role in evaluating the trees that they grow up, because we're not going to have genetic markers for everything. But the more information that we can give them for their toolkit, the better off they'll be, because then they can plant trees that are more likely to have desirable traits and be winners versus planting a bunch of trees that they're going to ultimately have to pull up because they taste bad or they look poorly or they don't have the disease traits that they want. And so I'm always interested in continuing to evaluate apples for new traits and to continue to acquire more genetic data so that we can improve the accuracy of our genetic mapping as well.
I love this whole idea of the aromas specifically because you mentioned this in the worksheet, Carsten, is that when you go to the Pometum, you can, have all of these really diverse experiences. And I think it's easy for, it would be easy for researchers to, just really focus on very practical things like, drought or cold resistance or disease resistance or, how can we just make them grow faster so that we can get more out there. But it is really important to...
I also think that the aroma has maybe been neglected during breeding of new cultivar, because what counts is the yield and the storage, that you can keep them, have a long shelf life and so on. And when you then go out in this Pometum and look at all these old cultivars, maybe they are not very productive, all of them, but they have their own special aromas, and there are so many different tastes you can experience when you eat all these different types of apples. And so I think it's a very important resource, and it will be really good if we can get more diversity on the shelves in the supermarkets concerning these traits.
Yeah, definitely.
I've got 3 questions left for you. So the first is, if people want to learn more about this kind of research, where can they go to learn more?
Well, the lab that I'm a part of at Dalhousie University, we keep a website updated with all of our most recent publications and also a little bit about the apple orchard that we maintain here, which is another diverse collection of apples located in Canada. And so they can visit that website at cultivatingdiversity.org. And that would be a good place to find more resources on the apple collection that we have here, where we're doing similar kinds of work in terms of genome-wide association studies for fruit quality traits.
And you also have your own personal website, is that correct?
Yeah, so I also have my own personal website, zoemigicovsky.com, and there's apple and grape resources there as well. And I'm always sharing interesting apples that I find in our orchard on my Twitter account. So feel free to follow me there and tweet me any apple questions you might have.
Totally. That is a dangerous door to open to me, because have so many questions.
Yeah, so that's Zoë Mig, so ZOEMIG, that you can find me on Twitter.
Absolutely. We will include links to all of those resources, as well as, I believe, Carsten provided a website for the Pometum, so people can check that out.
We have what we call an Apple key. It's also in English, so you can go in and look for about 300 different cultivars and read about them, what we have in the collection.
Is that where people can also order their branches if they are a home grower?
There is actually, yes, at the Pometum, there is also a place where you can order them. Yeah.
I love that. Excellent. So then how can people, if they want to get involved with supporting apples or apple research, how can they get involved?
Well, one thing I could mention here is that we have actually a project here in Denmark which is a kind of crowd breeding. So connected to this apple collection, some years ago, I was not directly involved, but I actually participated in it. So I got then small bags of 10 seeds harvested on different cultivars. So we knew what the mother tree was, but actually we don't know the father. So they were free pollinated. So then you sow them out. So a lot of private people were then getting their small seedlings in their garden and have grown them up now for about 5 years and have apples on them now. So that's a kind of how we have involved people around in Denmark to breed new cultivars. Of course, it's not a very sophisticated breeding scheme, but it's a way to involve, normal people in doing something. So, I mean, everybody can, of course, harvest kernels from apples they eat and then sow them out and see what they get. The likelihood of getting a superior cultivar is not very high, but it's quite fun.
It's like an apple lottery.
Yes, it is. Yeah.
Zoe, do you have anything to add for that question?
No, I don't have any other specific recommendations. That sounds like a really cool program. I guess the only other thing I could say is continue to buy and eat apples. And I think, I guess also if you're buying and eating apples outside of the top couple apple cultivars, then you're helping to show that there's a interest and support for new apple cultivars. So not just Red Delicious.
Totally. So then last question. Well, actually, you know what? I have 2 questions for the last question. So what is your favorite way to eat apple other than raw? Like a family recipe, or just what is your favorite way to enjoy them?
I love apple crisp. I have been eating apple crisp almost every week for the last 2 months during harvest. So yeah, that's definitely my favorite, just with oats and brown sugar and cinnamon and baked on apples with a nice crunchy coating on top. There's nothing more warming in the fall. So that's definitely one of my favorite ways to do it. I don't have a specific recipe. I just throw the ingredients together every time. But if you have good apples, it always turns out pretty good.
Yeah, I also like apple cakes and different things. But one thing I would like to mention here is maybe that one of my colleagues, who is also author on the paper we have published here, is doing a lot in wine making. And one of the things he's doing cryo wine, or we call it ice wine. Maybe you know this expression. So you concentrate the apple juice by freezing it down and letting it melt slowly, and then you get a very concentrated apple juice, which you can then ferment not just to a cider, but to a quite high percentage of alcohol. And these ice wines or cryo wines, they are really good. So that's the kind of new product we are doing here.
Wow. I have never heard of that before. That's amazing. All of those sound delicious. Now I'm hungry. Yeah, so that was sub-question. And then final question for real this time. What is one fun fact that listeners would not know about you if all they had was your research?
So the one that I put in the worksheet that you gave out was that I love to travel. And so one of the cool things I've done while traveling was hike the Inca Trail to Machu Picchu in Peru.
Nice. Carsten?
I have a sister living in Greenland, and I have been there a couple of times. And there's absolutely no apples in Greenland because there's almost no trees there,
Well, wonderful. Maybe Zoe can fly out and meet you sometime and combine a little bit of both.
Oh, yeah.
Yeah, that sounds lovely. Well, that's all the questions I have for you guys today. I have learned so much, and it's just been such a delight to have you on the show. So thank you for your time and joining us.
Thank you, too. It has been very nice to be with you and talk to you.
Yeah, thank you so much for having us.
Absolutely. Our pleasure. Thank you for listening to Field Lab Earth. You'll find a link to today's paper and other resources for this episode in our show notes or on our website. If you have any questions, comments, or recommendations for show topics, please contact us at podcast@sciencesocieties.org or on Twitter @FieldLabEarth. If you'd like to hear more content like this, please subscribe, and don't forget to rate and review us on Apple Podcasts or anywhere else you find your podcast if you like our show. This podcast is a joint production of the American Society of Agronomy, Crop Science Society of America, and Soil Science Society of America. Special thanks to Lobo Loco for the use of their song Spook Castle on the intro and outro of our show. Opinions and conclusions expressed by authors are their own and are not considered as those of the American Society of Agronomy, Crop Science Society of America, Soil Science Society of America, its staff, its members, or its advertisers.